I'm very excited. It's our 20th session, and I'll hand it over to Sadamani to welcome everybody to this exciting talk.
Thank you, Pavitra. Good morning, everyone. Outside In is a forum. It's a series of webinars that we have been taking part in since the lockdown began, almost since June. And this is being carried out in the theme of ecology. So to begin with, we had a series of lectures by different eminent people in the area. And now we are expanding on this and trying to keep to one theme per month. And my co-host, Dr. Deepa Agashe, and I have woven together a theme this month on microbes. So hope you will enjoy the session. And I would now like to request Dr. Pratibha, to introduce the speaker.
Thank you, Professor Sodamni. Hi, everyone. I am Pratibha, working as postdoc at NCBS, and I'm really thankful to you all for joining us today. And also thankful to the organizers and especially to Deepa, who have given me this wonderful opportunity to introduce our final speaker on this session on microbes. I am very happy to welcome Dr. Sujal Phadke from Rady's Children's Institute for Genomic Medicine, United States. Sujal had been studying her doctoral and postdoctoral studies, trying to understand the genetics and evolution of sexual reproduction in microbial eukaryotes in context of ciliates and fungi. She has done her PhD from University of Houston, Texas, and she carried out 2 postdoctoral studies at Duke University and University of Michigan, United States. Then she joined as an early career researcher in J. Craig Venter Institute, where she tries to incorporate her expertise on sexual reproduction in microbes into some insights into understanding on human genomes, which I'm hoping that she will touch upon today in her talk. So I'm really excited to have Dr. Sujal Phadke today and looking forward for your talk.
Thanks, Pratibha. Indeed, it has been an exciting journey. I should say that when I joined a PhD program, I was broadly interested in evolution. I didn't know I was going to start working on microbial mating systems. This is something that I stumbled upon and found really cool. And so I followed upon it. And I think that's one of the lessons of PhD program. They let you do that. They let you get hooked onto a question and just dive into it deeper and deeper, or a set of questions. And you get to really study the system as much as you want. And in the process, you also actually learn, scientific methods. So all of that was, although unexpected, it was very rewarding. And so hopefully I will share today with you what I have learned and sort of convey the coolness of microbial mating systems. I'm not working on microbes right now. So my current job at Rady Children's Institute for Genomic Medicine is actually sifting through human genomes to find the causative or most likely causative mutation for rare genetic disorders. The reason I stumbled upon this was because I was working on a specific microbial mating system, and I will talk just a little bit about it at the end of the talk. So keep that in mind. So with that, I have sort of divided my talk into 3 stories, and then I will give a general introduction that kind of ties up the stories and gives you a little bit of taste of what you're going to see, and then I'll elaborate on each of those aspects in each story. So we should get started with the introduction. Very basic stuff. Since there are high school students in this audience, when I was a high school student, I didn't know what was a mating system, for example. I hadn't thought so thoroughly about it. So here is what it is. What is a mating system? It's basically how a species manages its sexual reproduction. It includes everything from how individuals look for partners, which partners they choose, why they choose those partners, how much time they spend looking for the partners, how much time do they actually spend in sexual reproduction, what happens after sexual reproduction. So you get offspring, how are the offspring's phenotypes, what are their genotypes, what are the consequences of sexual reproduction. So it's a mating system is really, everything around sexual reproduction of a species. And there are a few terms that you will actually encounter quite a bit, and they could be confusing. So I have tried to give a meaning of each, and I'll try to provide a little context. So the word gender, we know, all of us know that word gender. And when somebody says gender, we quickly think about males and females. Well, of course, males and females are the most recognized genders, but more and more in humans and in other organisms, not just microbes, but also in metazoans, people have started realizing that male and females are sort of 2 points on a spectrum, right? But that's what gender refers to. It's a spectrum, and there are these two definite points on the spectrum. How well defined they are, it depends on the species. The other word you will hear is sex. And this word, if you start working on sexual reproduction, can confuse you a lot. So it can mean different things in different contexts. One, it can mean just male and female. So, sometimes when you fill up those forms, paperwork, you have to do a check mark for sex, and it says either male or female. In that context, it is referring to as gender. It can refer to the process of sexual reproduction. So exactly all of the aspects that I was just talking about, the courtship behavior, who chooses whom, so on and so forth. In a very specific context, sex is defined as meiosis and recombination. I'm not going to go into details of what is meiosis or what is recombination, but these are events at molecular level that are hallmarks of sexual reproduction. So if you're looking at an obscure species, and if you want to know whether they're having sex or not, they're undergoing sexual reproduction or not, this is what you're looking for. You're looking for whether there are signs of meiosis, where you can see signs of recombination and meiosis. And this mostly comes in the context of evolution of sex. So a lot of people for a lot of years have tried to study evolution of sex. It's a topic in itself that I'm not going to get into, but suffice to say, evolution of sex is an enigma. It is an enigma because asexuality offers a lot of advantages that sexual reproduction doesn't, at least in the short term. And so there is a whole field of people studying evolution of sex, and mostly in that field, sex is referred to as meiosis and recombination. So there are these different meanings to sex, but especially if you work on microbes, you'll come across a term called mating type quite a bit. And I use the word mating type and sex interchangeably because it kind of functions the same way, and I'll talk a little bit more about it. But mating type is basically a morphologically indistinguishable gender, okay? It's a type that determines who you mate with in a population. So that's your mating type, except that in a species that has no sexual dimorphism. So in humans, there is, at least to an extent, there is some sexual dimorphism. There are many species with sexual dimorphism, but in microbes, mostly the sexual dimorphism doesn't exist. And so for the microbes, we tend to call it a mating type, although I'm going to use the word sex and mating type quite interchangeably. Okay, the other thing I wanted to cover in basics is, do microbes have mating systems? And of course, the answer is yes, otherwise I wouldn't be here talking about it. But I want to actually make clear what I mean by microbes. And what I have here is a tree of living organisms. Here you have prokaryotes, which are archaea and eubacteria. You may have heard bacteria undergo conjugation, that is, kind like sex. I'm not talking about that. I'm going to focus on all these rest of the species, or many of these rest of the species that are eukaryotes. At the bottom here, we have metazoans. As humans, we are part of metazoans. Our cousins probably are fungi, closer to us than other species here. Anything that is not a metazoan, not a fungus, or not a green plant are termed protists. Protists are polyphyletic, and it is these protists that I have spent much of my time studying mating systems of. A lot of protists are microscopic. And when I say microbe, that's what I mean. Microbes are polyphyletic. You find microbes in all of these groups, but they're only united by their microscopic size. So if you bring me anything microscopic, I will be very excited and I will look at it. And that's the sense in which I'm using the word microbe. A lot of people refer to microbes as just prokaryotes, but I'm going to be mainly talking about microbial eukaryotes. Many microbes do have mating systems, and it is not always easy to determine whether a microbe is having sex or not. And so in the next slide, I'm going to give you just a tiny bit of taste of what are all the ways people have taken to actually figure out whether something or some species is having sex or not. Because sex is pretty important too. I mean, sex gives you a lot of advantages through meiosis and recombination. It generates variation that is so important for the whole process of evolution. And so sex is important. And so people are interested in who is having sex and when and how. So how do you know if microbes are having sex? When it comes to metazoans, it's fairly easy, I would say. You could, watch them. You could sort of catch them in action, so to say. With microbes, it's not always possible. There are some microbes that you can do direct observations on. So, for example, in many fungi, you see that the sexual stages look very different from non-sexual stages. So usually fungi go through a life cycle where they have an asexual stage, then environment changes. Usually a lot of microbes have sex when they're starving or when in like nutrition depleted environment. And so if the environment changes, you see this shift towards, development of sexual stage, and they basically undergo meiosis and recombination. So in fungi, these stages look different. And in some cases, they look so different that actually taxonomists have confused them for being two different fungi. And I encourage you to go on the internet and just look for these examples. They are just pretty amazing. It is quite a bit different. In some cases, like algae, it's pretty easy to observe, at least some algae, it is pretty easy to observe gamete fusion. You can just go collect a sample. If you're lucky, you will actually find a sexual stage in that sample. If you look under the microscope, if you give it right conditions, you'll see some gametic fusions there. When none of this is possible, and when you have fancy equipments like electron microscopes, you look for ultrastructures. So every sexual stage, and I'm going to show you an example of this, but it produces these, for example, in fungi, it produces these spores that are very particular to that sexual stage. You don't get those spores, you don't get these morphological structures unless that species has had sex. So you're looking for these morphological hallmarks under a microscope. And so you can look for that. Like I said, you can change the environment, and you can just collect wild isolates and see if they have sex, see if you can find fusion or not. So these are all direct observations, but as you can imagine, these are all only applicable to the ones that you can either collect and keep alive in the lab, or you can culture in the lab so that you can keep studying them. That's not true for many species though. Most microbial eukaryotes, most protists, we haven't figured out how to culture them. So either we luck out by collecting them, which doesn't happen very much, or we have to find indirect ways to figure out whether they are having sex or not. And so there are indirect markers of sex, which are basically hallmarks of recombination. My postdoctoral advisor, Dr. Heitman, and among others, especially in fungi, I have done quite a bit of this work, but there are many others who have studied these hallmarks of recombination in microbial eukaryotes. There are some very cool studies where people have figured out another way, another indirect marker of whether sexual reproduction is occurring or not. And I'll encourage you to go and read the literature by Dr. Longston. He's in Iowa State University, I believe. And he has figured out, he has basically a set of genes that are meiosis specific, meaning these genes are only being expressed, they're only working when the organism is undergoing sexual reproduction. And so what he could do is he can mine the genomes of these species. It's easier these days to sequence a genome than to culture and organisms. So you can mine genomes of these species for the presence of these meiosis specific genes. You can get even fancier. You can actually not just look at the genome, you can look at the transcriptome, which means you look at the mRNA level. And you can say not only are these genes present, but these are also being expressed differentially, for example, in asexual versus sexual stage. So these are indirect markers of sexual reproduction that you can also use. And people have used these to, place on this tree, for example, where sexual reproduction is happening and where we still lack evidence. There are species in which we lack evidence, but that doesn't mean they're not having sex. It just might mean we haven't stumbled upon the evidence. So now that I have introduced you to how people actually go about studying sex in microbial eukaryotes, I'm going to start with my stories. The first one is called Many Choices. And I'm going to start with the norm. Most of us grow up studying that there are two sexes, male and female. We also grow up studying that sexes are self-incompatible, which means that we need both the sexes or parents of both the sexes to contribute in order to, reproduce successfully. And that's not wrong. That's pretty much what happens for sure. So you have two sexes in many species, whether it be humans or, as I have shown here, A and alpha, much less cuter. But A and alpha are the sexes that you find in something like yeast species, for example, baker's yeast, what we use to make bread or beer. So A and alpha are male and female, pretty commonly known sexes. But what if I tell you that there is an organism called Dictyostelium discoideum? It's a social amoeba. You may have heard about it. It's pretty cool even otherwise. But this thing has three sexes or three mating types. They don't look different from each other. So, that's why they are called mating types. But there are distinct three groups in the population and they mate with each other. One mates with two mates with three, and one also mates with three. So there are all these possible pairwise combinations that are possible. If that's not crazy, how about Tetrahymena thermophila? This was the organism that I actually studied in my PhD. This is what got me hooked onto, studying mating system. This thing has 7 sexes, and they all mate with each other. So every sex has like 6 choices. In yeast, for example, there are 2 sexes. You have, A and alpha. Alpha mates with A mates with alpha. It's like, okay, that's my choice. But in Tetrahymena, you have 6 sexes to choose from. That's pretty cool. And so I wanted to know why, I wanted to know how, and I wanted to know so what. So I started reading more and more about this. And I think the first story, I'm going to cover some part of what I did in my doctoral studies. So let me just tell you quickly where this organism lies in the eukaryotic tree of life. It's a cousin to actually malarial parasites. So epicomplexan is the clade for malarial parasites. Ciliates is where Tetrahymena is. It's pretty commonly found around the ponds, around fresh water bodies. And so this is the habitat that I have shown here. If you really go to any nearby lake, any nearby ditch, and you collect some samples and look under the microscope, it's likely that you're going to find it. It is also culturable, which is really good, which means you can enrich for it and see more of it. So this is a ciliate. It's a free-living ciliated protist, not an organism that whole lot of people care about. But it's cool because when we studied how many species of Tetrahymena have how many sexes, we found quite a bit of variation. So what this tree shows is 2 clades, well, 3 clades. The top one is an outgroup. Then the middle one and the bottom one are the 2 clades of Tetrahymena. These are all species of Tetrahymena that you find across the US, worldwide, for example. And when we looked at how many sexes these species had, they had quite a bit of variation. It ranged from some species have 3 sexes, some species have up to 12 sexes. Tetrahymena thermophila, which I showed you earlier, which has 7 sexes, isn't even the limit. There are some species that have 12 sexes. So what are these sexes doing? We didn't know what to do with all the other species. So we decided that we are going to focus on Tetrahymena thermophila, and we are going to study it a little bit more in detail. This is not something that you get to see quite a bit. Why would different species have different sexes? So what we did was we asked why. What advantage is having 7 sexes over 2 sexes? And this is something that I touched earlier a bit. 2 sexes gives you, not really a choice. Sexes are self-incompatible, so A has to mate with alpha. Alpha has to mate with A if they want to produce offspring sexually. In 7 sexes, you have quite a bit of choice. So let's ask who mates with whom and how frequently. Do they all prefer each other the same, or do they have preferences? So we did that. Good thing is, like I said, Tetrahymena is culturable, and people have also developed genetic tools for it, which means you can sort of paint these sexes differently, and you can observe them in a petri dish. Now remember, this is a single cell organism. The sexes don't look different, okay? So you need some sort of way to say that in a mixed culture, you need a way to tell sex 2 different from sex 4 and sex 4 different from sex 5. And so we chose triads because it was much more manageable. Out of the 7 sexes, we chose 2, 4, and 5. No particular reason other than the strains were readily available. We mixed them in a petri dish in equal numbers. But before mixing that, we tagged them. We didn't actually paint them. We used genetic markers. And so what I have color-coded here are genetic markers that are drug resistance markers. So sex 2, we gave some mutations to it that were resistant, that made it resistant to a drug called 6-methylpurine. So on and so forth. So each sex had a different drug resistance marker. So even though they looked the same, we had a way to track them separately. Among 3 sexes, you can expect 3 types of pairs, either 2 mates with 4, 2 mates with 5, or 4 mates with 5. So these are the 3 types of pairs we were expecting. And I have posted an actual picture here. It's kind of really cute. In a petri dish, when you take these sexes and you basically starve them in order for them to start having sex. And when you starve them, they form these little heart-shaped pairs. And that's how that's how you can visually tell that, oh, look, they're mating. Otherwise, if a cell doesn't find a partner, or for whatever reason it's not mating, it is, the bottom cell here, it's not mating. It's a single cell right there. So, but you can get a lot, like large populations mating in a tiny petri dish, and then you can pick these pairs, and you can actually look at the drug resistances of the progeny. So what happens in these organisms is they mate, they form pair, they exchange their genetic information, and then they separate. So it's like a renewed cell that is also an offspring. So you don't actually get, different cells, different offspring. You get 2 renewed cells that are now new in their genetic makeup after they have exchanged genetic information with each other. So for example, here, the pair between a sex 4 and a sex 5, sex 4 is tagged with cyclohexamide resistance, sex 5 is tagged with perimomycin resistance. When these 2 mate, they exchange the genetic information, and the cells after mating, they now have resistance to both of these drugs. So you can tell a single drug resistant means this is a parental cell that has never been mated, but a double drug resistant is a cell after or formed after mating. And so because you have tagged them with separate drug resistant markers, you can actually, literally pick these pairs and put them in these, expose them to these drugs. Only those that have the drug resistant marker will survive, and others will die. So then you look at, okay, how many pairs were formed between 2 and 4, 2 and 5, and 4 and 5. And the expectation is here is that if there is no met preference, if they all, like each other the same way, you kind of expect a random distribution. You expect equal number of pairs being formed, since you started with equal numbers of them. But if they have a preference, and I have given an example of what a non-random mating would look like, or a preferential mating would look like. Let's say 2 and 4 like each other a lot more than either of them likes 5. So you'll see more pairs in that dual drug resistance treatment than others. So this is a very clear assay that will tell you how many pairs were formed between which sexes. And so we did that. I don't know what I was expecting, what would have made me more excited, whether I found a preference or whether I didn't find a preference. I think that's a cool part about science that I got to learn. No matter what outcome came out, it was going to be cool. So to give you the conclusion, there was no met preference. So what we did was we did that experiment with between 2, 4, and 5, but we also did it in other triads. So we substituted 7 for 5, we substituted 5 for 4, and we substituted 4 for 2. So we did this experiment 4 times, each time with a different triad. And we looked at how many pairs were formed between which sexes. And what the x-axis shows you is the combination, the pair combination or pairwise combination. And the y-axis shows you the mean proportion. And so the dotted line is basically the expectation that you have when there is no preferential treatment or preferential, mate preference there. In pretty much every treatment, we didn't find a preference. You do see some skew in the triad 2, 5, and 7. That's the third panel here. And it looks like 2 and 7 don't really like each other. 2 and 5 maybe like that more. Later on, we conducted experiments where we figured out that that's not actually driven by the sex itself. The strain that we used for sex 7 was a bit of sick, had some other issues with it. So it was just dying more. So it wasn't just available as much. So in a nutshell, we didn't really find any preference. It would have been cool, we could have asked why is, something being preferred over the other. But here it's even cooler. I mean, think about how do you even make a system where you have these 7 sexes that all like each other just the same? How does that work at the molecular level? Why does that evolve? Unfortunately, I don't know the answer to that. But it's a very cool thing that one can explore, maybe with these other species or using some mathematical or theoretical approach. But to give you just a quick summary, what we do know that mating systems in ciliates are highly diverse. I only gave you an example of Tetrahymena and the species in Tetrahymena, but we actually looked at number of sexes across ciliates, and ciliates are these super diverse organisms. And just the same, the diversity in the number of sexes, how sexes are inherited is just mind-blowing. And so this was not only restricted to Tetrahymena, we found it across ciliates. And so the conclusion there was, yeah, they're highly diverse and we just don't know why. But there are other things that we don't know. We don't know why and how does the perfect self-incompatibility evolve. Is it also true? Does it also apply for other species? Or there is preferential mating in other species? I mean, who knows? I graduated at that point and I didn't conduct these experiments after. We also don't know why multiple sexes. And this problem, this question has been asked by many, many times, and there are theories associated with it. Not a whole lot of experimental research, I would say, but it's also hard to perform. And so I will just give you a quick look into why this problem is interesting. So I have a table here. In the first column, I have the number of sexes. So if a species has 2 sexes versus 3, 4, up to 9, I didn't go all the way after 9. Then the pairwise combinations that you would expect. So this is, the middle column is just straight away combination problem. You have 2 sexes, you pick 2 of them, how many pairs you get. So in 2 sexes, you get just 1 pair, A mates with alpha, alpha mates with A, that's 1 combination. In 3, you get 3 combinations. So in our experiment 2, 4, and 5, you had those 3 pairs. If you go down those columns, it will tell you how many combinations you expect. And of course, as the number of sexes go on increasing, assuming perfect self-incompatibility, as in everyone mates with everyone, you get those combinations. But when you look at the third column, it is the population that is available per sex for mating. So when you have 2 sexes, and if you are sex alpha, you have like 50% of population that is available because that's sex A. And usually sexes are in, 1:1 ratio. So there is, roughly half of alpha, half of A, or half of males and half of females. So in two sexes, you have 50% of population to each sex available for mating. In three, it goes to 66, 4, 5, it goes on increasing. But as you notice, the amount of population or the percentage of population that is available for mating for a particular sex is a case of diminishing return. So it's nice that, oh, I have more population available for mating, which means I can, put my genes, I have higher chances of putting my genes into the next generation because I just have higher chances of mating, everything else being equal. But as this advantage that newer sex gives you, as the number of sexes increase, this advantages, it basically diminishes more and more. And so there's going to become a point where you don't have that much more advantage by adding a new sex to the population. Maybe if you're less dense population, it's already hard to mate or find mates, then maybe in those populations you have more sexes. All of these are hypotheses. We don't really know the reason, but this might be one of the reasons why these species are increasing the number of sexes, or they're not consciously increasing, but evolution is driving the increase in the number of sexes, because as we know, tetrahymena and other protists as well, they have very less dense populations as well. So that might be one of the reasons. But there are many unanswered questions. And so just as I left my PhD with many unanswered questions, I'm going to leave this story here and move on to the next one. But this is a problem that many people are interested in, and hopefully there will be a way for us to figure it out. So I'm going to go to the next one. It's called One is Enough. So the context here is that we learned that sexes are self-incompatible. We learned that there are mostly two sexes, male and female, A and alpha. Those are the most commonly known sexes. But we also learned that many species have many sexes. So, why many? Why two? And can one be enough? Can you have sexual reproduction with just one sex? Apparently you can. Any question that you have about is this possible, usually there is going to be a microbial eukaryote that has made it possible. Figured out a way. So Cryptococcus is one such fungus. It's microscopic. It's a fungus, so it's not a protist, but it's a microscopic fungus. So it's a yeast actually, and it's a cousin to the baker's yeast. It's usually found in eucalyptus trees, associated with bird poop. So it's present around in natural habitat. This was my postdoctoral research, and the reason people are actually studying this fungus is not because of its cool sexual system, or that's not how it started. It started because this was creating outbreaks in different parts of the world. I have a picture here from the United States. This is the west coast of the United States, and between around 2000 to all like that first decade, Cryptococcus created havoc in many regions of the Pacific Northwest of the United States. It was these meningitis outbreaks that people were succumbing to, and people just didn't know what's happening and what's causing it. And once they figured out Cryptococcus was causing it, they started doing research on, so what is making this possible? What could be the reasons that this has suddenly started expanding? What the right panel shows you is the distribution of Cryptococcus sexes. And this is where the story gets interesting. If you look, so this is not from the the same region as the outbreaks, but this is an example, a representative picture that you also see in the region of outbreaks. What you see here, and there is a lot of detail here, but what I want to draw your attention to is that MAT alpha. That's the gene, or that's the allele that determines, if you have that, your sex is alpha. The other one is A, MAT A, and if you have MAT A, your sex is A. So it's A and alpha sex, and if you look pretty much in every box, MAT alpha or sex alpha is the predominant sex. And it's true, no matter whether you look at clinical isolates or go around and sample eucalyptus trees, for example, and isolate this yeast, most of the time, and if you look at the mating type, most of the time you are going to get sex alpha. So that was curious. There was no reason that there has to be a link between this distribution of sex and outbreak. But, in basic research, you study anything that is interesting, of course. So what we did was we looked at what is this sex alpha doing? What is it doing in population? So we brought it to the lab and we, grew it up in the environment that it likes to both grow itself and also undergo sexual reproduction. So this is a reminder, traditional sexual cycle is between A and alpha. And what happens to this yeast is when there are two sexes, it changes, it starts out at yeast and the two cells mate, and then it starts differentiating, and this hyphal stage that you find, spreading around the colony of the yeast, that's how that's your visual marker of, oh, that means the cells in that colony underwent sexual reproduction. And if you take this colony and you put it under the electron microscope, for example, you get to see these ultrastructures that are meiotic spores. And these meiotic spores are the ones that are formed only after this sexual reproduction. These structure, this stalk, which has a very complicated name as all things fungi, but this stalk over which you have this little spore, these are the hallmarks of sexual reproduction in this fungus. So what we did was the isolates that we studied, they were capable of having this traditional sexual cycle. So if there was another opposite sex that we gave them from somewhere else or a lab isolate, it had no problems mating. But what we noticed was it didn't need that. It didn't need that opposite sex. When we put these isolates of alpha or sex alpha by themselves without the opposite sex in a conducive environment, and remember I said most microbial eukaryotes, and definitely fungi, when they starve of nitrogen, for example, or just generally starve, they start having sex. And so when we put these isolates of alpha by themselves in this conducive environment, lo and behold, they started forming hyphae, which made us think, oh, wait a second, what's going on? And so when we looked at the ultrastructures, indeed, there were lots of meiotic spores there. And so it seemed like, oh, wait, it has figured out how to have sex by itself. Doesn't even need the type A. And what's important is two things. One, it is present in eucalyptus trees, in many habitats, but it's still not as dense. And so I believe Cryptococcus also faces the same dilemma. It's like it wants to mate, it wants to, sex is good, but there are no mates. So, that might be an evolutionary pressure for this unisexual cycle to evolve. And the other point I want to make is when you have this traditional sexual cycle, you have A and alpha both contributing their alleles to the offspring. And so you get spores of both mating types. When you have alpha, you're mating with alpha itself, you only produce alpha type spores. And we basically, the current hypothesis, the explanation to the geographic distribution of these sexes is that there could be this unisexual cycle happening in nature where you have alpha mating with itself and producing alpha spores, and that's what you get. And so this was an important point in trying to establish, at least trying to explain the geographical distribution. I by no means was the first one to explain this. In fact, my postdoctoral advisor, Dr. Heitman, have done a lot of research on this. But the strain that I studied was actually the strain that was creating havoc in healthy individuals. Joe, who was my postdoctoral advisor, had already shown this similar thing for a different species that preferentially causes infection in immunocompromised patients. This isolate that we studied was doing it is a primary pathogen. It also infects healthy people. So this was pretty exciting to be able to, find this evidence of unisexual cycle in there. And so this was one of the other points I wanted to make in things like yeast, for example, where you don't really get to see a whole lot. You always need a microscope. You always are looking for ultrastructures, but you can look at genetic level and you can study this sexual cycle a bit more. So what we did was there's quite a bit of genetics known about sexual reproduction in this species. So we decided that we are going to target a few genes and we are going to delete the genes that are, are negative regulators of this pathway that leads to sexual reproduction. So A produces a pheromone, alpha produces a pheromone, each has a receptor for the pheromone of the other. And so this is a pheromone response pathway. And so there are negative regulators for this pathway, there are positive regulators for this pathway. And so if you delete the negative regulators, you are kind of enhancing the pheromone response, and you should see, earlier or more hallmarks of sexual reproduction. And that's what we saw. What you see here is the edge of the colony, of the yeast colony in the WT, which is wild type. We didn't see in that environment, we didn't see, a lot of sexual reproduction going on at that time or at the given time. When we deleted the negative regulators, we saw hyphal formation and sexual reproduction taking place. When we overexpressed the positive regulators, the same thing happened. So this was our way of figuring out, okay, they're having sex with themselves, but is it the same? Are they using the same pheromone response pathway? And at least it seems at least partly true. So it's like a rewiring of genetic machinery to, figure out a way of how to reproduce sexually when you don't have your partner. So what we know is in Cryptococcus, one sex is enough for sexual reproduction. And we saw the morphological as well as, other hallmarks. I didn't show you the meiosis and recombination, but Joe has done work on this as well. What we don't know is how much does unisexual reproduction, the sex by itself, is contributing to actually these pathogenic isolates. I mean, it is easy to hypothesize that, okay, you have this alpha mating with alpha producing only alpha progeny, but is it really contributing to the outbreak? It is indeed true that clinical isolates are of sex alpha, but right now this is just a speculation. There are some studies that have looked at the virulence of alpha versus A, and there is some evidence to that too, but there is a lot of things we don't know in this. So here, studying mating system in kind of an obscure fungus that nobody was caring about before the outbreak happened, and basic research into it could be and was very rewarding as well. So with that, I'm going to go to my last story, Two to Tango. This is my favorite. Others are too, but this is my most recent favorite. And so we saw that multiple sexes are present in many species. One is enough for some species, but it is true. There is no denying that two sexes is the norm in many species, at least many species that we can study that are culturable, that are visible, metazoans, for example. And so two is the norm in most studies so far. And so microbial eukaryotes have also, there are some that have stayed by the norm. And so this story is about another fungus. It's called Allomyces. So again, not a protist, but an early diverging fungus, a lot different from other fungi probably. It's not a yeast. It's a filamentous fungus. Fungi has this ridiculous diversity in the forms. Some are yeast, some are filaments. You also, of course, know the mushroom bodies that are formed. But Allomyces doesn't form mushroom bodies. It's not a yeast. It's just a filamental hyphal fungus. And usually if you, collect soil, moist soil especially, and bait it with, things like hemp seed, and look under the microscope, you should be able to grow Allomyces. It's present pretty commonly around. It's not a pathogen, free living sort of fungus. So I'm going to show you two videos here. These are unfortunately not my videos. I wish they were because they are really cool. In the first video, what I'm showing you, you see some orange structures and you see some white structures. And what you're going to see is, what you're going to see is the gametes being released. So these are the white gametes. They're slightly bigger, slightly larger. They're motile. They're being released and they're going around. And in a little bit, you're going to start seeing the orange ones being released. And this is Allomyces. So the white gametes that you see, we are calling them females because in many species, females are larger. And you see those orange gametes that are being released. They're tiny, they're many, but they're also motile. And after the release, they just all start swimming around. And when you zoom in, my postdoctoral advisor had sent me this video, and this really got me hooked. When you zoom into them, look at the white big gamete trying to follow the small orange gamete, I'm saying gametes because this species has been studied by many, and by now we know that these are, two different gametes of two different sexes. But once they are released, they engage with each other, and at one point you actually also see one of them being fused with the other. It's probably somewhere here. So these are Allomyces gametes, and this is a species, Allomyces microgynous, that has two sexes, that has two sexually dimorphic gametes. And so this is pretty much the norm. I mean, this makes you think of egg and sperm, which is what we study about humans or, other metazoans. Okay, my video, oh, there you are. So these are just a few snapshots to show you the coolness of this fungus, which is quite a bit different than the other fungus we were just talking about, Cryptococcus. It looks nothing like that. It's very different. So, the diversity in mating systems, even when you come out of protists, the microscopic organisms are just teeming with these diversity. So we are studying this species, this genus currently. We have genomes for quite a few. And one another cool thing about this is this sort of sexual dimorphism that you just saw, you don't see it in every species of Allomyces. There are some species where the gametes don't look different. They're all small, they're all the same size, they're all motile, and these are isogamous species. It is this moniliformis and neomoniliformis, this top clade that I'm showing here. But there are species that show this, other species and microgynous that show these anisogamous, sexually dimorphic gametes. And so even within Allomyces, there's this nice continuum, nice variation between, different species have different mating systems here. This is not my work, but I wanted to put it there to make a connection to the human genomes that I was talking about. And after this, I'll show you one quick specific about information about specific disorder on the internet. But these are two studies that make Allomyces even more desirable as a model organism. So in humans, we know that egg and sperm interact through a molecular complex called Caspar. These are sperm-specific complexes that are on sperm. And Kai et al. Looked at the presence of Caspar in many different species, and that's the top picture here. You, of course, have them in Homo sapiens, which is us, but you also have them in Allomyces, and that's cool. Allomyces have many Caspars, in fact. And not only that, there was a study done that was published by University of Minnesota, or scientists at University of Minnesota, and they had a pheromone or a hormone called cyrenin. And the story about cyrenin is pretty cool. Cyrenin, actually, the word comes from Greek sirens who used to lure their mates by singing songs. But anyway, the cyrenin, this is the function of cyrenin. It's a hormone that lures the gamete of the opposite sex. And what the scientists at University of Minnesota did was they had this synthetic cyrenin that they had, and from the Allomyces too, and they used that and they studied the action of that cyrenin on human sperm. And again, lo and behold, human sperm responded to Allomyces cyrenin and also the synthetic version of it. And so it means that the cyrenin that is working in Allomyces through the Caspar complex, it has some relatedness structurally or, by descent. And it has, conserved that function on human sperm as well. So what the bottom panel shows you was that what these scientists do is they expose these sperms to the cyrenin. And as the sperm is exposed to the cyrenin, the sperm starts moving and towards the gradient or towards the cyrenin. And you can actually measure this movement by using some electrodes. And this electrodes that's the action that you are seeing here, this voltage difference that you're seeing here. And you see that wherever there is cyrenin, the green line, and the green line and the blue line. The blue line is the control cyrenin. The green line is the Allomyces cyrenin. You see these human sperms, trying to swim to the cyrenin. when you apply that might be Fredo, I'm probably mispronouncing that name, but that's a sort of a deactivator of cytidine in the layman's term. When you take away this effect of cytidine, you don't see this movement. And so it's really cool that Allomyces has this cytidine that can not only work probably on Allomyces gametes itself, but also on human gametes. And so what we do know is Allomyces can provide a great model for studying anisogamy, for studying many aspects of sexual reproduction. If there are conserved mechanisms of gamete interaction between egg and sperm or two gametes of Allomyces. Sexual dimorphism, we know egg it's not motile, but sperm is. But in Allomyces, we saw that both gametes are motile. So we can study sexual dimorphism at many levels. We still don't know quite a bit about sex determination in Allomyces. So I just put it there about how does sex determination work, whether it's sex chromosomes, whether it is dosage dependent. There are many questions, and I bet there are going to be interesting answers to that too. So with that, I want to leave with these two memes that I made on a day when I didn't really want to work, but I didn't want to feel guilty for not working. And so I'm going to leave you with that. And I probably went over time, but if there are questions, that I will be happy to take them. Thank you.
Thank you, Sujal, for that wonderful talk. I would like to go first. One of the questions that I have, I was still wondering why in ciliates do we see a lot of sexes? Like what advantage do they have? Like there is no cost of maintaining those sexes because like if there is no preference, or is there any advantage like in some conditions these sexes are useful for them?
Unfortunately, I don't know the answer. I can speculate why they have more sexes. In terms of cost, so this is something that we did think about, like what is the cost of having a new sex? These are molecularly different, and we are just beginning to understand the molecular structure of it, what molecular events undergo during development of these sexes after sexual reproduction or expression of these sexes. We still don't know much in terms of the specific differences between sexes, but there is no reason to believe that they're differentially costly to produce. So I don't know how much cost plays a role into producing sexes. Also, I don't know how to really measure the cost of producing sexes or producing a new sex, for example. So unfortunately, I don't have a satisfactory answer for you, other than some evolutionary mechanisms that we
But
discussed.
it's so much interesting, like how many sexes they have, 16 in. So I have one more question from Dr. Sri Hari, who asked about this. I think the same question of why different having the advantage of having different sexes in the microbial mating. And if they have this different sexes, how does the progeny looks like? They carry similar parental genomics, like their genomes are similar to their parents?
Mm-hmm. It depends. It depends on which microbe we are talking about. The one that we talked about in Tetrahymena, for example, there is a mechanism. So the progeny, when the cells mate, they inherit from genetic information from both parents. Right after they separate, they become new cell. There is a mechanism to ensure that only one sex is expressed in the progeny, even though they have a possibility of at least two. In that specific case, actually, they have a possibility of all the 7, but I'm not even going to get into the details of the complex genetic mechanism. But there is a way for the cells to ensure that one sex or a progeny gets one mating type or one sex. So their sex is fixed after their sex, and then once they have sex again, their progeny will have different sexes and so forth. But again, it depends on what type of genetic or sex determination mechanism you have. In some cases, you have a genetic sex determination, meaning you inherit it from the parent. In some cases, you have stochastic sex determination, meaning that you don't inherit the sex itself or the mating type itself. What you inherit is a probability of having a certain mating type. And if it is not making sense, I will point you to literature by Dr. Orias at University of California, Santa Barbara. He has this cool study where he showed that how do celiacs inherit this probability rather than just, sex itself. And it has something to do with a single gene being able to produce different sexes with different probability. If that makes sense.
So one more question we have from Sukriti, who asks us, like, how do we identify the meiotic genes in different organisms?
So that's a good question. You have a set of meiotic genes that people have shown by studying different organisms that they are specifically expressed in meiosis. And the way to know that is you take an organism and you grow it at different life cycle stages. And you basically test the expression of, whatever genes that you want to test. And you look at exactly which point in life cycle is that gene being expressed. And these meiotic specific genes usually shoot up during meiosis. In terms of once you have this set of genes that are known to be meiotic specific, how to exactly mine genomes is you basically then you have a new organism for which you have a genome, and you have this set of genes that you want to, find in these genomes. You basically look for homology or sequence similarity by descent in these genes. So you take the sequence, the DNA sequence of these genes, and you compare it with the genome, and you see whether that sequence or a sequence that is similar to that sequence is present in this genome. And then you look at the structure of that sequence, the structure of the gene. A more foolproof would be to do transcriptomics, where you actually then not just look at the DNA level, but you can look at the RNA level. So not just what information is present, but how it is being expressed. And then you can look at whether those genes are not just present, but also being expressed or not. Does that make sense?
Yeah. So another question that we have is from Professor Sodamni. She asked that what if they start killing each other because there are with or without sex, like because there has to be balance between the sexes. So is there any competition between the sexes for the mating, or like behavior wise?
The competition part is the one that we addressed through that experiment where we, basically took away the competition and asked whether they still prefer one over the other, and they didn't. We didn't try different densities, but it didn't seem like they really compete with each other in the sense that there is no, at the molecular level, we haven't looked at whether one is producing more pheromone, because that would be one way to compete. In that species specifically, it is not pheromone mediated, meaning the two partners need to be physically interacting in order for them to, form a pair. So it's pretty hard to compete in that sense. So I'm just not clear on how they are going to compete with each other than just having different numbers in the population. That probably is the easiest way for them to compete.
Okay, so we have another two questions from Ayush, who he wants to know, like, what's the design of the experiment to suggest the mating types for the preference, and also wants to make sure that, and how do you make sure that it is not random or due to environmental factor, and whether it's the same condition that you see in the wild species.
So the design of the experiment. So what we wanted to do in that experiment was to ask, if given a choice, and if given a similar choice, meaning if one sex is presented with two choices in the same way, would you prefer one over the other? And in order for us to be able to not introduce some sort of bias, we had to be really sure to, count specific, number of cells of each sex and then mix them together. So they were all equally present or represented in the population. In terms of environmental effects, they were all in the Petri dish. They were all, mating each other. So we, other than controlling the initial numbers where we equalize them, we didn't control anything else other than putting them all together. So they were free to, move around. They were free to form a pair through mostly physical contact. So whoever they mate or find, a mate to pair with. In terms of environmental factors, this was a very controlled environment. So there is a specific set of conditions that you use to induce starvation. So they were all, exposed to the same conditions. In terms of how to ensure it is not random, well, we wanted the choice to be random in the sense that we wanted them to choose, and we didn't want us to introduce any bias. So I'm not sure I understand that part of the question much.
He wants to know, like, do we really see these similar kind of patterns in the wild species, not
We
in the
don't.
lab? Yeah, that's
Yeah.
not been explored yet.
Yeah, we don't know. The reason being that it's pretty hard to study these organisms in wild because they're microscopic. So you have to bring them to the lab and you have to, grow them to. So people have studied the distribution of these sexes in the wild, actually, by painstaking efforts. And earlier, before we knew anything about the genetics of this was even more, it was even harder. We do know that they are in skewed sex ratios, meaning not all of them are equally present in the population, just like Cryptococcus, where we see alpha is predominantly, in the population, and A, you rarely find it. In Tetrahymena, you have these 7 sexes in different populations. They have different, relative frequencies. So they don't all get, the same choices as they did in our experiment. But our experiment was more geared towards, are there any, inherent differences? If given a choice, if given, equal numbers, do they still prefer one over the other? And the answer there was no. But when you imagine a population that has skewed sex ratios or skewed number of sexes, I bet, there are, proportionate matings between sexes in the natural populations.
Thank you, Sujal, and it was a wonderful session today. So now I would like to hand it over to Professor Sudhamani to do the closing statement.
Yeah, thank you, Sujal, for a very interesting talk and talking to us about how there are multiple sexes in some microbial organisms, and taking us through the science behind it in a very simple way. She's from Rady Children's Institute for Genomic Medicine, for those of you who might have joined late, so you can keep in touch with her. I'm sure her science excites you all. I would like to thank Dr. Pratibha for moderating the session today. And especially, I would like to thank Dr. Deepa Agashe, who has been co-hosting this whole month with me on the One Nice team on microbes. And I also wish to extend my thanks to the communications team, especially Pavitra, for supporting this initiative, and also to NCBS. And yes, thanks to the audience, as always, for coming on Sunday morning like this. Thanks again to the speaker. Yeah, so next week, what we do is on 6th December when we meet at 11 a.m. The topic is going to be different. So we switch from microbes to fresh, those species which stay in fresh waters, like mollusk. And for this, my co-host is going to be Dr. Samir Padye from Biological Life Science. And the speaker on 6th December is going to be someone from ATRI, which is nothing but Ashoka Trust for Research in Ecology and Environment. And the speaker is Dr. Arvind Madhyastha, who is going to talk about shells and slimes, molasses in general. Do stay on, and we'll see you all next week. Until then, please take care.
Yeah.
Thank you.